Hi, everyone. Welcome to today's EMSL Learn webinar, where today we are going to talk about experimental design considerations for user proposals. The EMSL Learn webinar series is designed to provide information to the scientific community on resources available at EMSL for research in the biological, environmental, and computing sciences, I'm sorry. Carly, can you advance to the next slide? Our webinars are posted on the EMSL Learn section of the EMSL website, and we invite you to visit that section to see our past webinars. If you would like to stay informed on upcoming webinars and proposal calls, please sign up to receive emails from EMSL, or go ahead and follow EMSL on your social media or various social media channels. Jenna is going to provide the links in the chat. Now, for today's webinar, if you have questions for the speakers, please post them on the Q&A section of your Zoom windows so that we can answer them during our Q&A session that'll happen around 12:45-ish, or when all the speakers are done presenting. And Kelly is going to introduce today's speakers. So at this point, I'm going to turn it over to Kelly Stratton. Thank you.
Hi, everyone, and welcome. So we're excited to talk to you today about experimental design as it relates to the EMSL user program. So I'm Kelly Stratton, a data scientist here and the Integrated Research Platform lead for data transformations at EMSL. Joining me are two other data scientists, Moses Obiri and Damon Leach, with backgrounds in statistics and biostatistics, including other things such as experimental design, machine learning, omics data analysis, and R package development. So I will start things off with some of the motivation for putting on this webinar, and then talk about the different perspectives that we can consider when designing experiments. Then I'll hand things off to Moses to talk about experimental design principles and to walk us through some examples that were inspired by previous EMSL user projects. And then finally, Damon will talk about batches and power calculations, and we'll wrap things up with some time for questions from you all. So the large-scale research letter of intent template now includes this checkbox shown on the slide that lets the PI indicate whether they're interested in getting assistance from EMSL staff with statistical design of experiments or statistical analysis. And so we wanted to supplement that new addition with some detailed information about experimental design as it relates to the user program. And just sort of for general awareness, if you happen to check this box, or if you've already checked it for this latest round of letters of intent, what this is doing is simply flagging that LOI for people on the EMSL side as we review those letters of intent to make sure that we can connect the proposer with the appropriate EMSL scientists as they develop their proposal. So the goal of this checkbox and of the webinar is really just to help all of you and show you some of the ways that we as statisticians presenting the ways that we think about experimental design, and also to invite conversations about this aspect of user projects at the proposal development stage. So the more that these conversations about experimental design and the statistical analyses that will be performed happen at this early stage of proposal development, the easier it's going to be to hit the ground running once projects begin. And you'll hear this throughout the rest of our webinar today, but if you have questions, please either ask them at the end here or reach out to one of the IRP leads, any of the three of us, or the other EMSL staff that you might already be working with, and we can either answer those questions or get you connected to the right person to help. All right, so now let's sort of dive in. I, I like to think about experimental design from multiple perspectives or lenses. I find that it helps to make sure that, decisions that need to be made about the experimental design are made from sort of the most informed position that's possible. And I recognize that just because you might have a beautifully powered experiment with 250 samples doesn't mean that that's feasible, or that it would even address a relevant scientific question. But when the constraints and the options are all out on the table, we can better understand the trade-offs that are available to us and make decisions about the experimental design and the downstream analyses. So to highlight the different perspectives that we can take, I want to consider some of these experimental design properties and take a look at why these questions are of interest to domain scientists, statisticians, and reviewers. So what type of information are these different people coming at it from different perspectives actually wanting to get from the answers to these questions? So from a domain science perspective, these questions here in the middle pertain to the scientific impact of the experiment, how to procure the information that's needed to address the question of interest, right? What materials are needed, what instruments should be used, are the right things going to be measured in the right system, under the right conditions, and all of those types of things. From a statistical perspective, these same questions provide information about the type and the structure of the data that's going to be collected. So whether there's an important correlation structure in the data, what analysis methods are appropriate, and whether there's going to be enough statistical power to answer the question that's of interest. So if we pick one of these questions here in the middle of the slide, let's see, what about what things or parameters or variables are thought to impact the outcome? So from the domain science perspective, the answers to this question are particularly relevant to the materials that will be needed in order to perform the experiment, right? What type of growth media? What is the actual sample material that needs to be worked with? Is it soils, isolates, or something else? And from the statistical perspective, this same question is critical for understanding the properties of the data that will be collected, right? Is it binary? Is it continuous? Are there images? Do we have repeated measures that need to be accounted for, and so forth? So those questions in the middle mean very different things depending on the lens that's being used. And if we consider a reviewer's perspective, they're likely looking for some combination of those other two perspectives, right? They need to understand what the structure of the experiment is, just to sort of wrap their head around it, and to be convinced or comfortable that the right things are being measured in the right system and that the experiment and the statistical analysis that will be performed are appropriate for answering that question of interest. So the type of information and how these aspects are conveyed in a proposal depend on a lot of different factors, including the type of study, the science domain, and the complexity of the experiment that's going to be performed. So before I turn things over to Moses to expand on this by providing a framework and some examples, I just want to say that some of these things are going to be items that are defined by the PI in the proposal. Others are frequently conversations with EMSL staff during proposal development or even at project kickoff. And some of them, like I'll call out randomization, which will be talked about a little bit later on, might be happening behind the scenes. So the user might not necessarily be aware of it. And I just wanted to make that last comment, just in case you're thinking, oh my goodness, I had a previous project and I don't remember talking about randomization. It happened behind the scenes if you weren't aware of it. So I just want to preempt any worry about things like that. All right, Moses, I'll hand it off to you now.
All right. Thank you, Kelly. So, Kelly, yes. Okay, have control now. Yeah, so Kelly just walked us through some of the key perspectives to consider when designing an experiment. And we also wanted to highlight two different types of scientific studies that can often be mistaken for one another, right? So what is an experiment, and then what is an observational study? And so an experiment involves intentional manipulation of certain variables that we call independent variables to study their effect or their outcome. And then an observational study involves recording these variables, these response and then independent variables out of observing some natural systems of interest. And so the key difference here is manipulation. An experiment involves manipulation. An observational study does not involve manipulation, right? So, for example, if we measure microbial activity and then compare them across different treatment groups, then you have an experiment, right? But then if we just observe or measure microbial activity from sampling some soil from somewhere without any manipulation, then you are dealing with an observational study. And so the reason why we want to particularly highlight the difference between an experiment and then an observational study is that depending on what you are doing, the type of statistical analysis that you use, and then even the conclusion that you can draw from the analysis will be different. And so typically, an experiment, you can do causal inference, that is cause and effect inference. You cannot do so in an observational study setting. And then the type of analysis and then observational study, you have to account for certain confounding variables or new additional even outcome variables that may pop up in your analysis. So the analysis will be different. The conclusion you draw from the studies will also be different depending on whether you have an experiment or an observational study. And so, like Kelly said, for this talk, we want to quickly go over some of the things we have to keep in mind when designing an experiment. And so there are three core experimental design principles. And here I want to sort of rehash what Kelly said, that we are aware that EMSL scientists already incorporate these principles in their research. And so this is just a quick refresher to highlight how important these principles are. And so the first principle is randomization. And randomization comes in two forms. The first one is randomly assigning your samples to your treatment groups. And then the second one is then randomizing the order in which the samples are run. The main purpose of randomization is to even out any inherent human or machine biases that can be introduced into your study so that you can cleanly compare treatment differences, treatment across your groups. The second principle is replication. And again, here too, we have two types of replication. We have taking biological replicates and then taking technical replicates. And so simply, biological replicates are samples coming from the same treatment group. And then technical replicates involves taking repeated measurements from the same biological replicate. And the main purpose of replication is to be able to quantify any differences or noise or what we call variability that is not coming from your treatment. And so any noise or any differences that are coming as a result of the natural variability in your system or the way you measured your samples, you want to be able to quantify that so that you can then also cleanly compare any differences that may be coming from your treatment, which you are interested in. That the main goal of the study. And so any statistician will tell you that replication is very, very important. And the last principle is local control. And local control also takes two forms. We have balancing and then blocking, which I can say maybe batching is part of blocking. And so balancing means that you have the same number of samples across your treatment group. So you have the same sample size across your treatment group. And then statistically, doing so would ensure that you increase your statistical power, so that later on, Damien will speak to statistical power and then what goes into it. But you want high statistical power. And so to achieve that, you have to make sure that your sample sizes across your groups are all equal as much as possible. And so that is balancing. And then blocking means that you account for any other variables that you are aware of that are neither the independent variables or the response variables, but you know they can affect your response variable. So for example, if you are running a study and you know that temperature can affect your response variable, but it is not an independent variable that you are interested in, then you have to make sure that your experiment is run with constant temperature. And so these principles are key. And like I said, we know EMSL scientists already use these principles, but in case still you need any help with sort of incorporating these principles, there are people here to sort of help you incorporate these principles in your research. And so combining these principles that I just talked about and then the different perspective that Kelly also spoke about, we think you end up with these 3 key steps that we think should be followed for you to have an efficient design of experiment. The first step in any research start with you coming out with your research question, right? And so Kelly spoke about how in doing so, you have to make sure that your research question is, one, domain relevant. And also, a very important thing is that it can also be answered statistically. Not all research questions can be answered statistically. So you have to make sure that your research question can be answered statistically. And so in doing so, it means that you can identify key variables from your research question. You can identify what are the independent variables here. What are the response variables here? Are there any confounding variables? So it's at this stage that you also start to think of, okay, based on these variables, then what is the statistical model that can answer this research question? And then you jump to the second step. What will be the appropriate design for to be able to answer my research question? And so you pick your design, and then in picking your design, you then also apply the 3 principles that I just talked about. That is randomization, replication, and then local control. And so you apply your principles, and then you then go to the last step, which is then you identify a consistent protocol. And this step is very important. It all speaks to this reproducibility. You have to make sure that if you are not there, someone that comes and then follows your protocol would get a data that is consistent with the data that you got. So you define a consistent data collection protocol. And then once you have your data, then you make sure that you choose a statistical method that aligns with 2 things. 1, your research question, and then 2, the design that you have. So we think these are the steps that are needed for you to have an efficient experimental design to be able to answer any research question that you have. And so in the next slides, I will go over some generic two examples. And I'll start with a culture-based experiment. And so let's suppose that you have this research question here that you want to investigate microbial growth for different strains in different media conditions. So that is your research question. And so from this research question, you are able to identify the independent variable or factors, which are your nutrient media type and then your strain type. You identify the levels that you have for each. So in this case, I use 3 levels for the media A, B, and C, and then 2 media type. And then it's at this stage that you also choose your response variable. So for this generic example, I chose the response as the optical density to be measured over time. So we have defined our research question. We have identified the relevant variables of interest. And so the next stage then yeah, sorry about that. The next stage then involves coming out with a design. And so based on the fact that I'm dealing with just 2 variables and I have just, I think, 2 or 3 levels per each variable, an appropriate design will be a factorial design. A factorial design would allow me to be able to fit what we call a complete model. So I've chosen a design based on my research question and then based on the number of variables that I've been able to identify. And so for this example, I chose 5 replicates for each strain and then media combination. But later on, Damon will speak about statistical power. Typically, the number of variable replicates that you use should be informed by the available resources that you have and then the statistical power that you want to achieve. And we talked about randomization. Randomization is key. It's very important in any experiment. So for this example, let's say there are 3 plates available. So this is an example of how you can sort of randomly assign these 5 replicates per strain and media combination across the 3 plates. The key thing here is that you want to mitigate plate effects from your study. You are not interested in the plate. You are interested in whether strain and media affect microbial growth. So randomly assigning the replicate to the plate will then even out the effect of plates across your replicates. And then the third principle of local control, you keep in mind, are there any variables that you need to control for? So I have listed some variables here as example that if you think temperature, pH, and other incubation conditions that you can think of can affect microbial growth, then you make sure that you keep all those variables constant. And so here we've chosen a design based on our study question, and then we've also then applied the 3 principles that we've talked about to be able to achieve a design that is appropriate for our research question. And so once we have our experiment and then we run our experiment, for this experiment, we can then collect our data. I decided to use an experiment where you can collect data over time. And so based on the research question and then based on the design that we have, it then informs the kind of statistical model that you can use. And so based on this example, one model that you can use is what we call a repeated measures ANOVA, because we've taken optical density values over over time, or you can even use a growth curve model to be able to also answer your question here. And so in using a repeated measures ANOVA, you can fit a model where you have an interaction between media strain and time, and then you plate as a random effect in your model. That model, based on your design, should be able to answer the research question that we stated earlier. You can also use a growth model curve like we have here, to then answer any question relating to whether there are differences between your strain or there are differences between your strain and then media combination. Yeah, so that was an example for a culture-based experiment. So now let's look at a soil-based experiment. So let's say you want to here study, and this is also another generic example that we came out with, study the impact of soil disturbance on plant growth. So again, here, based on the research question, we are able to identify the independent variables here, which is your treatment, whether you are controlling the plot or you are disturbing the plot, and then the sites. I use 2 sites here as an example, but we've identified the independent variable, and then you identify your response variable. So you can choose any variable pertaining to plant growth here, whether the biomass, height, or yield. But the key thing here is being able to identify these variables from your research question. Once you do that, you then come out with a design. You just come out with a design based on your research question. And so for this research question that you have, I decided to use what we call randomized complete block design, where we are randomly assigning the treatment across the different plots in the 2 sites. And so that is our randomization strategy. I think we use 8 plots. I use 8 plots here per site, which means that you have 4 replicates per treatment combination. And so we've taken care of the replication aspect of the principles of experimental design. And then you also account for any confounding variables that you think may affect plant growth, such as soil moisture, nutrient levels, and any external factors that you think can affect plant growth. You have to make sure that they are constant across the 2 sites that you are then performing your experiment. So you've done that. The next stage then coming out with then a consistent data collection protocol. You collect your data. So in this case, you measure your plant height after a few months, and then it comes to then choosing the appropriate statistical model. And so based on our research question, based on the design that we use, that is a randomized complete block design, an appropriate model would be, for example, would be a random effect ANOVA model, where you model the plant growth as a function of treatments, and then you have plots nested within sites as a random effect. The key thing here is to account for the plot-to-plot spatial autocorrelation that will be present, because we know that plots coming from the same site will be correlated. And then you also want to account for your site effect. And so this model allows you to do that. And so like I said, this model is linked to our research question, the design that we chose, and then the data that we collected. So having done that, this is also a generic example that I chose, data that I simulated. So you can then show a box plot to then look at whether there are differences between your 2 treatment groups, in this case control, and then whether the plot was disturbed or not, and then you can then draw your appropriate conclusion from it. And so here, I've talked about this a lot. There is sort of a link between your research question, the kind of design that you use, and then the kind of statistical analysis that you choose. And so knowing this at the early stage of your research will save you a lot of headache later. And so knowing this means that, like I said, at the stage where you are thinking about your research question, you are able to get some idea about, okay, what design would be appropriate for my research question, and then what statistical method can I also use, right? And so for the two examples that I spoke about, there were examples which involved comparing two groups and also repeated measures. And so we know that based on these examples, and then based on the design that we chose, we can use repeated measures and ANOVA linear mixed model to answer the questions that we wanted to answer. And so the link between your research question, experimental design, and then the statistical method that you use is very, very, very important. And so we've listed a couple of different research questions here, and then the appropriate statistical analysis that you can use depending on the question that you have. I will not go over this, but the key thing that we want to say here is that if you are at this stage, or if you are at any stage of your study, and then you want help with either the planning or the design or linking your statistical method with your appropriate data that you have, you come talk to us, and then their expertise here at EMSL to help you with that process. And so I talked about using replicate, and then how replication is very important in the statistical analysis and then in the experimental design framework. And then the key part of replication is calculating how much sample size that you should use. And so what goes into that is statistical power. So you have to be able to know how much power that you want to achieve. And so I will hand it over to Damon. Damon will speak to how to do statistical power, and also one other key aspect which involves batching, which batch effect, which is also a key part of the principles that I talked about. So over to you, Damon.
Awesome. Thanks, Moses. Yes, so I go to the next page. Awesome. So there are just a couple other considerations that we kind of wanted to talk about when it comes to experimental design and statistical analyses. And one of those is this concept of batch effects. And in the context of omics data, batches are going to be the samples that are prepped or ran on an instrument in distinct groups. And this is typically separated by time. And so this often is due to logistical requirements, such as the number of samples that we're able to run within a workday, or the fact that the samples are being collected over a long period of time, and they need to be run on an instrument before all the samples have been collected. So all of this leads to scenarios which we have then batches, which lead to then batch effects. And the batch effects are going to be defined as these non-biological differences that are occurring between the samples ran in those different batches. And the reason that this is such a big issue when we are working with omics data is that these batch effects can mask the biological features. And so when we're running these statistical analyses, we're finding these differences, but they're only caused by the batch and rather than by the biological kind of question of interest, we're not really being able to answer the questions that we're trying to answer. So for example, if we look at this figure here on this slide, we have these samples that are colored in red, and those correspond to batch 1. And then we have these samples in blue corresponding to batch 2. And what we can kind of see is that the median log2 abundance in those batch 1 samples do in general seem to be larger than the medians from batch 2. And so if this is a scenario where we have these batch effects, we might get these differences, but it might not actually be telling us anything important with our data as it pertains to the biology. And this also here highlights the need for proper randomization strategies, because, for example, if we had an experiment that was comparing these treated strains of an organism with control samples, and suppose we put all of the treated strains in that first batch and then all of the control samples in that second batch, we would be dealing with this correlation between batch and the biological signal. And we wouldn't necessarily be able to parse that out as well as if we had kind of completely randomized the data and we had some treated strains in both of the batches and some of the control samples in both of the batches as well. And so luckily, when it comes to batch effects, we do have a lot of different ways that we can handle and correct for batch effects. But first, I just want to point out that some instruments, simply because of how they function, are just more prone to batch effects. For instance, if multiple batches of samples are being ran on an LC or GC mass spec for relative quantification of peptides, lipids, and metabolites, we often recommend looking into kind of whether batch effects may be present. Here at PNNL, we've actually developed an R package called malbac-R, which compiles 12 different batch correction algorithms that have been created by scientists all across the country and kind of put them into one package so that way users can kind of quickly and easily be able to try a couple different ones, see which one makes the most sense for their specific data set. And with that being said, some of these methods do require a bit more forethought when it comes to the experimental design. And this is just because some of the methods require some additional information. For example, some of these samples do require quality control samples to be placed into the batches. Others require internal standards to be spiked into the samples. There are, however, a wide variety that don't require any of those requirements and thus have fewer restrictions when it comes to kind of what batch correction methods can be used on the data set. Within this package, we also do supply some methods that are commonly used for assessing batch effects. Some of these are going to be a bit more qualitative, such as kind of analyzing the clusters of samples using a PCA plot, but we do also have a quantitative measure that's been included in the package as well. So we can kind of see here with this figure is that on the top, we have this data set where there are 3 different batches. And in this PCA plot, they're very heavily clustered by batch. So there's no overlapping with any of these data points. All of the red ones cluster together, green together, and blue together. But after I believe we ran combat with this data set, all of those circles now kind of are more heavily overlapping, and we are able to notice a bit more of the biological signal that's happening with the 3 different treatment groups that were applied into the data set. The second topic that we wanted to highlight here as just kind of part of the other considerations is this concept of statistical power, which has been kind of talked about a bit throughout this presentation, but just to kind of give it a bit more of its own kind of segment. Power is defined as the ability to statistically detect a difference when a difference truly exists. And so typically this is going to be in the context of determining sample sizes. We want to make sure that we have enough samples to ensure that our experiment does have sufficient power to detect what we're trying to detect. And so when we're doing a power analysis to calculate the sample size per group, we need the 4 following values. So first, we're going to have the desired power, and this is typically going to be set to 0.8 or 80%. We're also going to have to specify the alpha value, and this is going to be the level of significance in the experiment. Typically, this is set at 0.05, suggesting that a p-value under 0.05 is going to be considered statistically significant when it comes to our results. We also need to specify the effect size, and with omics data, this is frequently going to be the fold change value of interest, and then finally, the standard deviation, and this is going to be the variability in our data. Typically, the standard deviation for a power analysis is going to be estimated from previous experiments that works with a similar data set. Oh, went too far back. So then to highlight how these different 5 parts are interconnected, we can state this following sentence. So assuming a level of statistical significance of 0.05 and a standard deviation of 0.49, with 20 samples or 10 per group, we have an 81% chance of detecting a fold change between the two groups. And this 0.05 is going to be our p-value, 0.49 is the variability of our data, 20 is the total number of samples in our experiment, or 10 per group, indicating that we have two groups in this experiment. 81% is then going to be the statistical power we've calculated, and 2 is the effect size. So we can also look at, and then another example using this heat map to kind of illustrate some of these interpretations. So here we have a few of the parameters that are just kind of set and stay constant throughout. So we've set alpha to be 0.05, the fold change as 2, and the standard deviation as 1.13. We then can present a range of power values calculated from the possible sample sizes for the two groups. So when we have 3 samples in each group, we then have a power of 0.21, which means that with 6 total samples, we only have a 21% chance of detecting a significant difference when it actually exists, which is a lot lower than our desired 80%. And for this specific data set, if we increase that number of samples to 10 in each group, or a total of 20, that power then increases up to a 0.47. So this is showing that if we increase our sample size, we are going to be increasing our power. If we went to 20 samples per group, we'd see an even larger power as well. So on the left here, we have the exact same heat map that we were just looking at, but then on the right now, we have power results that we would see if we increased the fold change that we'd like to be able to detect, increasing it from 2 on the left to now 6. So now we see that when we have 8 samples in each group, the power for detecting a fold change at 2 is going to be this 0.42, but the power to detect a fold change when the fold change is 6 is now going to be that kind of gold standard of 0.8. So this kind of highlights that holding the other things constant, the sample size, alpha, standard deviation, we have more power to detect a larger effect size. And one way that I kind of like to think about it is, suppose we were on a safari and we were looking for animals of different sizes. The animals of different sizes are kind of like the effect size. So if we don't see an elephant, there likely isn't an elephant there. And we have a decent amount of power when it comes to that kind of conclusion. However, at the same distance, if we don't detect a smaller animal like a bee, we can't be assured that it isn't there. We have less power to detect something that's going to be the size of a bee. And that kind of carries over then into this concept of the effect size. Similarly, we can also look at the impact that standard deviation has on our power calculations. So if we keep everything constant, but we decrease our standard deviation from 1.13 to 0.879, we are able to increase our power. Keeping the group sizes at 8 each, it jumps from 0.42 to then 0.8. So this demonstrates that the more variable that our data are, the less power we have to detect the same fold change. And this is important because since different data sets can have different amounts of variation in them, this means that just because we ran a power analysis on one data set that concludes that we need 5 samples per group, this may not be the same for another data set. We may need more samples or we may need less. And so when it comes to these power analyses, the biggest thing is that it's always going to have some type of trade-off, because it's very common that resource constraints are going to require us to prioritize something when it comes to experimental design. For example, when it comes to designing an experiment, we may need to consider and ask the following questions. How many samples can we actually afford? Can we reduce the scope? Perhaps we might need to have fewer conditions. Can the question of interest be reframed to leverage a more efficient statistical technique? So for example, if we need 16 total samples, 8 from each group, we could achieve the goal power of that 80% or 0.8 power. But if we decrease that to only needing 12 total samples, the power decreases to 0.72. And it may be that this reduction in power, this 0.08 reduction, is deemed reasonable given all of the logistics and resources that would be needed to acquire that number of samples or to run them on an instrument required for the study. So this is just kind of to highlight the fact that more samples isn't always necessarily better, and it does highlight the fact that when it comes to experimental design, it is a very big balancing act of what we can and should be prioritizing. And so finally, we would just like to emphasize that the EMSL staff are here to help you, even during proposal development. And just thank you. We're happy to answer any questions at this time. And you can always reach out to us or the other EMSL scientists that you may already be working with if you have questions specifically about your proposed research.
All right. Carly, can you take down the screen share right now while we do the Q&A? Awesome. All right. Well, thank you, everyone. Fantastic presentations. And we do have some questions from the audience. So go ahead and we will launch into those. The first question, which I think he will appreciate being first, is I think it could be for you, Kelly. When we hear back regarding our large scale, or when will we hear back regarding our large scale research LOIs or letters of intent? If invited to submit a proposal, will we be matched with EMSL scientists to help develop experimental design for our proposals?
Yeah, so notifications are going to go out on March 4th, and those will include suggestions for EMSL staff, EMSL scientists for you to contact to continue working on the proposal if you're invited to submit one. Yep.
Fantastic. Okay. And this question is for Moses. I think it refers back to your presentation. How did you arrive at 5 replicates as optimal for the culture experiment?
So in that example, we just chose that number at random, but like Damon went through, typically that number would depend on the power and other things calculation that you do. And so that number will depend on the power that you want to achieve for your study and then the amount of resources that you have. And we help with that here, like Kelly said. And so if you need help, you can come talk to us. Yeah.
Okay. Damon, for you, what batch correction method should I use?
Yeah, that's a good question. That definitely depends on the data that you're working with. It's definitely not a one size fits all. That's why we definitely highlight in the Malbac R package that there are a couple different ways that you can kind of quantify how well your data set is working, whether that just be kind of looking at different PCA plots or the kind of more quantitative function that we've applied. Yeah, it's going to be definitely data set dependent. So the best thing is just to kind of try a couple of different options. And that's why it's good to know what your data looks like before you kind of run the experiment. So that way that gives you more options to look at when it comes to batch correction. So if you already have the quality control samples in the data, then that's another option that you can look at. But if you don't have them, you can't consider those ones then.
Okay, fantastic. Another for you, Kelly. What if we have questions about designing an experiment that doesn't look like either of the examples or any of the examples that were presented here?
Yeah, if that's the case, definitely reach out to one of the IRP leads, or if you're already working closely with another scientist at EMSL, and they can help get you connected to the right people. For the examples for this webinar, we were trying to pick sort of broadly applicable things, so it's not surprising that there might not be something that fits exactly with that.
Mm-hmm. Okay, another for you, Moses. My study involves a lot of variables, like more than 20. Can I still use experimental design?
So yeah, thank you. So that is a great question. And that is a question that typically people use as an excuse for not using an experimental design. And we must say that no matter the amount of variables that you have, from 2 to 1,000, there are experimental design options available. If you are dealing with high dimensional design, there are design options that involve space filling designs and adaptive designs that are available. And so once you run into that, make sure that you have someone to talk to that can help you with that. So yes, the short answer is yes. There are many design options available for high dimensional designs that can help you with any study question that you have.
Great. We have a few more minutes left and a couple different questions. So we do have time for another couple questions. Audience, if you have anything burning that you would like to ask, I would say please put that into the chat now so that while you've got everyone's ear here, it's really the best time to ask questions and learn more. So next question, Damon, for power calculations, how do you know if the standard deviation value is, how do you know what the standard deviation value is that should be used?
Yeah, that's definitely going to be the hardest parameter probably to determine when it comes to the experimental design and power analysis setup. The biggest thing that we usually use is just previous experiments. And so the goal would be to use a similar data set to the one that you're trying to run. So maybe it's like the same organism or the same kind of treatment groups. And yeah, that's the kind of the best thing is just to look to the past. What other data sets have we ran? What other analyses? And then we can use that as a proxy then for this calculation. Because yeah, obviously if we haven't ran the experiment yet, we don't know what the specific standard deviation is for this current one.
Right. Yep. Okay. Allison is asking if both EMSL and JGI offer the capability that we need, which facility should we request from? Kelly.
Yeah, so I guess I would say that that's probably a good thing to talk through with the scientists from EMSL and JGI, and they're going to be able to help you understand sort of what the different benefits might be to going with one versus the other for your particular situation. So I don't have a sort of hard and fast answer on that one, but yeah, I would recommend talking to them.
Likely somewhere in the omics fields, you know.
Right.
Okay, we have another one from Marina. So for longitudinal studies, like 2 years of metagenomics data, are there any tips for designing our sampling strategy to improve statistical power? Does anybody want to take that one? Do you want me to repeat the question?
Do you want to take that? Yeah, so I can take that. So if here, it will depend on your research question here. And so if we take it that you are doing just a longitudinal repeated measure study, then there are sort of questions that can be asked to help you arrive at the things you need for you to improve your power, right? So for example, how many repeated measures are you taking, right? What is your sampling interval? Are you taking measurement every 2 months, 3 months? How many measurements do you want to take every sampling point, right? All those things are important. So there are things that you can do. I would say that there are things that you can do to increase your power. And then those things will depend on how you want to structure your design here. The things, the sampling intervals and how many samples that you want to take at each sample.
All right. Again, if anybody's got more questions, go ahead and drop them in the chat. I've got another one from Allison again. Can we request the Eberlite support in the current EMSL JGI call? Kelly, do you know the answer to that one?
I don't know the answer to that off the top of my head, so we'll have to find the answer and get back to you on that one.
Yeah.
Yeah.
And so, Allison, we will email you and let you know. the answer, unless there's others who want to know too. All right. We have come to the end of all the questions that have been submitted. And we're 5 minutes from finishing. This might be a record to finish early. uh,
Linda, sorry to jump in. I wanted to let you know I do have an answer to that question from Allison that came from one of our project managers in the user program. The eBER Lite is available through the advanced photon source in the FICUS call.
So it is available.
It is available.
Fantastic. All right. So, oh, I
There
think,
are more questions.
yep, I think I just saw a couple pop in. Okay, so Bo was asking, I may have missed a slide, but how do I calculate the power using the sample size, alpha value, standard deviation, etc.? How to calculate the power? Does anybody want to take that?
I was just gonna say there is like an actual algorithm. We actually for this presentation specifically kind of left it out just because it can be a little bit wonky and not the most intuitive. But yeah, so if you need the help with the calculations, there is a specific algorithm depending on kind of the specific experiment of interest. It is probably always easiest just to ask someone from like a statistician, just because there's a lot of nuance when it comes to kind of the specific experiment and just slight little tweaks. I don't know if Kelly, if you had anything else you'd want to add with that.
Yeah, I mean, there's a lot of tools online, but I would also echo Damon's caution that depending on the experimental structure, it would be pretty easy to pick something that was slightly different than what you intended. So if you have questions though, any of us can help point you in the right direction or help with the calculation.
All right. Well, yep, I don't see any more questions being asked, so I think we're gonna call it good. Thank you everyone for joining us today. And just to let you know, our next webinar is going to be on March 12th, visualizing live cell dynamics and metabolic activity through chemical probes. So if you are interested in live cell interrogations using chemical probes, this is the webinar for you. We will see you there. Thank you, everyone.